Alright, in this video we're going to talk about cell division and the actual process of it because we've been talking a little bit about cell division already. And the first thing you need to think about are chromosomes. Chromosomes are basically all of your DNA bundled together into a little strand complex. Normally in eukaryotic cells, so human cells, it's found in the nucleus. But in prokaryotic cells, it's just found floating around the cell. This right here that you see is actually the chromatin of the E. coli. And yes, E. coli is that bacteria that makes you sick when you eat chicken, that's bad and all of that. But E. coli is actually really good for science because it has such a distinct chromosome that we can cause changes to it and do a lot of genetic testing with the E. coli. These chromosomes are actually prokaryotic cells and prokaryotic chromosomes. So that's how they're found. They're just found floating around the cell. When we're talking about eukaryotic chromosomes or human cells, so anything that's eukaryotic is human, animal, plant, all of this. Things with a nucleus. You should remember that. With nucleus. They have a lot more DNA than prokaryotes. Prokaryotes have much less. The human DNA has about 4 billion base pairs. Normal prokaryotic DNA can have even as little as 10,000 base pairs of DNA. So basically, when we talk about DNA, you'll know a little bit more about what that is, but prokaryotic cells have maybe 10,000 times less DNA than eukaryotic cells sometimes. This is not always, obviously. Now, in its natural state, when it's just floating around the cell, DNA is in the state of chromatin. And chromatin looks something like that. And chromatin is a complex meaning just two things mixed together. Let me just write that down. Mixed together of DNA, and DNA is obviously this double strand that you've seen a lot of times before, and a few proteins called nucleosomes. Those proteins just hold the DNA in little chunks and help make the chromosomes have the shape that they normally have. Humans normally have 46 chromosomes. There can be changes in that, but that causes problems. And we'll talk about a few of those in a little while, but why are they even packed into chromosomes? And that's for the accurate division of DNA. When the cells divide, you have to have the same amount in both of the daughter cells, and to do that, DNA condenses itself into chromatids. And they look something like that. Normally, chromatin, which is the state that DNA is in, looks like this, all squiggly and nasty. And kind of weird. It's like spaghetti, a bowl of spaghetti, or like if you've seen a bowl of soup with a lot of noodles, that's what it looks like. Then, during the process, which we'll see down here in the cell cycle, it kind of condenses into these little X's. And you'll see when we study the cell cycle that little strands come and pull these two little sides apart so that you're left with something that looks a bit, let me get a color that's close to it, more like this. And they'll be stuck in two little pieces, and each one of these goes to a daughter cell, or if you want to say it better, one of the two cells that come out of one. So these each go to a daughter cell. Daughter one, I spelled daughter wrong, one, and then this one goes to daughter two. Now, how do the cells actually divide? They are actually broken into two different types. Prokaryotic cells divide very differently than eukaryotic cells. Actually, very differently would be an exaggeration. It's just much more simply. Prokaryotic cells divide by something called binary fission. And you can see it written right here, binary fission. And binary fission basically means, if you break down the words binary, that's two, and fission means to break apart. So it breaks apart into two. And it's broken into two basic steps, and all cell division is broken into these basic steps. The first one, DNA replicates. So you start out with one normal cell with its prokaryotic chromosome, and then it duplicates. A little thing runs around it, and it makes one ring into two little rings. Then these two rings each start moving off to the sides. They start moving this way, this one starts moving that way, and it starts splitting. When the cell starts splitting, a little indentation starts forming like that. This is actually, and you guys are probably going to laugh, called the cleavage furrow. And yes, just like the cleavage you're thinking about. Cleavage furrow. And it starts pinching in. This little piece that you see right here starts coming in deeper and deeper until it starts pinching in, and it fully pinches in into two separate cells, two identical separate cells, each with its full strand of DNA. So it has full DNA and all of the required parts and stuff. So it has all of its organelles, each one has a full set of everything. And that's how binary fission happens. It's super simple. DNA, it's broken into two steps. DNA replicates, and two cells divide. Now,